Welcome back to my YouTube channel. In the course of this video lesson, I'll be solving different practice questions that cut across different topics in biology. Let's get into the first practice question, and it says, seed plants are divided into, we were given options. How do we solve this question? This question is extremely very easy. When we see a question in any subject at all, be it biology, chemistry, physics, use of English or mathematics, it is very important for us to understand the topic to which that question is from. And from the look of things, this particular question is from the topic in biology called classification of living organisms. I know you want to ask, how did I know it's from that topic? I saw a very important word in this question, and the word was plants. Are you there? But now they are specific. They said seed plants are divided into. That's what the question is asking, and we are giving options. This question is very easy. Now, it is very important for me to explain to you the concept of classification of plants. Remember, I said that this question is from the topic in biology called classification of living organisms. But since the question is dealing with just plants, I'll be talking about classification of plants because plants forms part of living organisms. Remember, living organisms are broadly divided into three groups, which are plants, animals, and microorganisms. So right now, we'll be talking about plants generally. So it is important for us to understand the overview of plants before we can even go ahead to solve this question. Now, moving further, it is important for us to understand that plants forms parts of the kingdom of classification. You know what we call hierarchy of classifying living organisms. It starts with kingdom. After kingdom, we have what we call phylum, class, order, family, genus, species. These are the hierarchy of classification. But this must be noted. Whenever we are discussing about plants classification, remember I said that the hierarchy of classification starts with kingdom. After kingdom, we have what we call phylum. But whenever we are talking about plant classification, phylum should not be there, but rather it should be replaced by what we call division. So in plant classification, it starts with kingdom. After kingdom, now division. Are you there? Remember, in animal classification, it should be kingdom, phylum, class, order. Are you there? But in plant classification, it should be kingdom, division, class, order. I believe you understand. I'm going to show you here on the screen so you see what I'm talking about. Moving further, it is very important for us to understand that we have various kingdoms of classification. Are you there? We have various kingdoms of classifying living organisms, and they are 5, which include kingdom Monera, kingdom Protista, kingdom Fungal, kingdom Plantae, or you say kingdom Plants, and kingdom Animalia, or you simply say kingdom Animal. So now you can see we are about stressing on kingdom Plantae because in this question I saw plant, but specifically what type of plant? Seed plant. So before we even get into talking about seed plant, it is very important for us to understand the concept of plants. Are you there? Now, moving further, it must be noted here that plants generally, listen carefully now, plants generally are broadly divided into 2 groups. Are you there? We have what we call the tracheophytes. We call them the tracheophytes. All right. And also we have another division of plants which are called atracheophytes or tracheophyta. Are you there? So these are the divisions of plants. Remember, I told you in plant classification, phylum should be removed and we replace it with division. That is why I call these two guys division of plants. If I was talking about animal, I'm going to call them phylum of animals. I don't know if you understand what I'm saying. So that's how it works. So first of all, plants, they are divided into two groups, majorly which include tracheophytes and atracheophytes. Now this must be noted about the tracheophytes. Tracheophytes generally, they are the vascular plants. Are you there? What do you call them? They are the vascular plants. They are plants with vascular bundles, which include the xylem and the phloem tissue. For what now? For getting nutrients from the soil, not just nutrient water, all right, to nourish the plants. So you must know here that tracheophytes, they are the vascular plants, whereby atracheophytes, they are the non-vascular plants. Are you there? So when they are non-vascular plants, it means that they don't have the vascular bundles in them. All right. What are these vascular bundles? I said they include the xylem as well as the phloem tissues. Are you there? So moving further into talking about the vascular plants, which are also called the tracheophytes, they are broadly divided into two groups as well, which include what we call the pteridophyta, all right, and also what we call spermatophytes. Are you there? So these are the groups under the vascular plant, the pteridophytes and the spermatophytes. Are you there? So moving over to the atracheophytes, which are the non-vascular plants, they include two other groups, which include what we call the talophytes. Listen, we call them the talophytes, and as well as the bryophytes. Yes, talophytes and as well as the bryophytes. So it is important for us to know examples of organisms belonging to these groups. So moving further, listen carefully. Now the way it works is this: organisms belonging to any of these groups, their name should end with phyte. Are you there? So organisms under the talophyta, what do we call them? We call them talophytes. Are you there? Organisms here, bryophytes. Organisms here, pteridophytes. Organisms here, spermatophytes. Are you there? So this must be noted. So you can see we've established on all of these things. You guys saw plants. I have to explain to you the concept of plant classification, which I just did, but there are more things to know. But listen, the first question we ask ourselves: what are seed plants? What are seed plants? These are plants that reproduce via their seed. Seed plants are plants that reproduce via or through their seeds. Are you there? So this must be noted. We'll see where seed plant actually belongs to in the classification of plants generally. But now note this, all right? Now it must be noted here that spermatophytes, they are broadly divided into two other groups. Listen carefully. Spermatophytes are broadly divided into two other groups, which include the angiospermatophytes, all right, angiospermatophytes, and as well as the all right, gymnospermatophytes. Are you there? So organisms belonging to these groups, we call them angiospermatophytes and as well as gymnospermatophytes. Are you there? I told you their names will end with phyte. The organisms there, examples of organisms there. So listen carefully. So now let's understand something. Actually, this must be noted. It must be noted here that the spermatophytes, they are the seed plants. So basically, if I don't call them spermatophytes, I just call them seed plants. Are you there? So this must be noted. I believe now you are trying to understand the answer to this question. I called spermatophyte seed plants, and they are basically divided into two groups, which are Angiospermatophyta and Gymnospermatophyta. I believe now you are getting the picture about the answer to this question, because we already said that seed plants are plants that reproduce via their seed or through their seeds. Are you there? Now you can see the answer to this question, because spermatophyte gave rise to these two. So what becomes the seed plant in all of these options? Is it monocotyledons or and dicotyledons? No, it's not the answer. All right, but it's closely related, but it's not the answer. I've not talked about this, so that is not the answer. Option B, is it Talophytes and Bryophytes? No, Talophytes and Bryophytes are classification under plants, but they are not seed plants. Are you there? Talophyte, Bryophyte, basically they are non-vascular plants. They are not seed plants. Are you there? So moving over, Tracheophytes and Fern. It can't be this, because Tracheophytes are broad groups of plants. Option D, Angiosperm and Gymnosperm. Wow, that is the answer to the question. So the answer to this question becomes option D. I believe you understand how this question was determined, how the answer to this question was solved is very simple, but I'm still going to further explain some concepts, so listen carefully. Now, moving further, we have seen that spermatophytes are seed plants. Are you there? And they gave rise to Angiospermatophytes and as well as the Gymnospermatophytes. Alright, so what's the difference between Angiospermatophytes and Gymnospermatophytes? Both of them, listen, they are seed plants, alright? They are seed plants. But now, here are the difference between these two groups. Angiospermatophytes, listen, Angiospermatophytes, they are also regarded to be called flowering plants, whereby Gymnospermatophytes, they are also regarded to be called non-flowering plants. Are you there? Now, what's the difference between them? Now, another difference is that for Angiospermatophytes, both of them are seed plants, they have seeds. But for Angiosperm, their seeds are enclosed. But for Gymnospermatophytes, their seeds are naked. Their seeds are opened. Are you there? So this is the slight difference between this group of organisms under spermatophytes. Are you there? So we have examples of Angiospermatophytes, which I'll be listing here in this video lesson. For Angiospermatophytes, they include corn, roses, lily, beans, sunflowers. Are you there? So these are examples of the Angiospermatophytes. But for the Gymnospermatophytes, they include what we call conifers, they include what we call cycads, they include what we call jingles. So this must be noted about this group of organisms under the spermatophytes. Moving further, let's quickly talk about Talophytes and Bryophytes. Alright, though we've gotten the answer already, but it's important for us to understand the concept of Talophytes and Bryophytes. Talophyte, Bryophytes, they are non-vascular plants. They don't have vascular bundles. They don't have xylem and phloem tissues. So that is why they live in moist environments. They live in environments where, or they survive in environments that are damp with little water. Are you there? So that's why we say that they live in moist environments. What are examples of Talophytes? They include algae generally. Are you there? Algae generally are Talophytes. What are these examples of algae? We have what we call the green algae, which is a very good example of organism under the green algae. It's called Spirogera. Remember, Spirogera is a green filamentous algae. Are you there? We have other examples of green algae, which include the chlorella. Chlorella and Spirogera are examples under the green algae under Talophytes. Are you there? We have other examples of algae, which include the red algae. So generally, Talophytes, we have three examples of algae, which include the green algae, red algae, and brown algae. For the red algae, they include what we call the rhodophytes. Are you there? Red algae, what do we have there? Rhodophyta is a good example of organism under the red algae. And also we have examples of the brown algae, which include what we call kelp. Kelp, K-E-L-P. Alright, kelp is a good example of a brown algae. We have other examples which include fucus. Fucus is also an example of the brown algae. So all of these must be noted. Moving over, we have other examples of Talophytes which include leaching. Leaching basically is an association between two organisms, which are algae and cyanobacteria. Algae cyanobacteria forms an association called lichen, but it's spelled leaching. Do you understand? It's pronounced lichen. So all of these must be noted. So you can see examples of organisms under Talophyta. So let's take it again on Talophyta. We have algae, we have leaching. Do you understand? Now basically here, algae are three, which include the brown, red, and green algae. So that is for Talophyta. Bryophyta basically include organisms like moss, MOSS, liverworts, and onwards. Are you there? All of these are examples of organisms under Bryophyta. But for Pteridophyta, they include fern. You can see fern here. Fern is a pteridophyte. Are you there? And Spermatophyta, we've talked about that. So I believe now you understand the concept about plant classification. All right. So let's get into the next practice question.
To have more understanding about chemistry calculations and solving all of these type of questions, I already have them on the chemistry masterpiece, okay, which is calculations and keynotes in chemistry. So if you are preparing to write the Jamb exam, this book is very, very important for you. Now, for more information getting the chemistry masterpiece, do well to direct message me via my WhatsApp number. It will be on the screen.
The question says, which of the following can cause shrinkage of living cells? All right. Option A, deionized water. Option B, hypertonic solution. Option D, hypotonic solution. And option D, isotonic solution. All right, this question is extremely very easy. Now, let's use an illustration to actually get the answer to this question. So let's say we have a container containing and all of these solutions, like different containers containing all of these solutions. So basically, the answer to this question is option B. Now listen, for a cell to shrink, either that cell is a plant or animal cell, it must be placed inside an hypertonic solution. So first of all, it is important for me to explain to you what all of these solutions are and how they work. All right, now listen carefully. Now, we said plant animal cell placed inside hypertonic solution. What happens to them? They will shrink. All right, for something to shrink, it means that it loses some part of itself. Do you understand? It's logical. Let's say you take a balloon and inflate that balloon. So let's say that balloon now, you've removed the air inside the balloon. You can actually use your hand to squeeze that balloon and it will shrink. But when it was inflated, can you just shrink it like that? No. So for shrinkage to occur, there must be something removed to have that in your mind. So now let us analyze this. Now let me just tell you this key point. So now let's explain. What is an hypertonic solution? An hypertonic solution is a solution that have higher concentration of solute. Remember, a solute is made up of solute. Remember, a solution is made up of a solute and a solvent. So this solute now in this case can be salt, can be sugar. Do you understand? So these are examples of solute that can be used in all of these solutions. So there is solvent, which is the liquid, which is water. And you know, cells generally is made up of over 70% of water. Remember, you know that cells are the basic functional structural units of life. So inside our cells that we have many organelles inside the cytoplasm of that cell, they are made up of over 70% of water. So have that in your mind. So the water will be the solvent. Do you understand? So let's get this straight. What's an hypertonic solution? So let's say this is the container containing an hypertonic solution. All right, and this is a cell, for instance. Do you understand? This is the nucleus of the cell. Let's say this is the nucleus of the cell. So basically, you know, this nucleus, this empty space, they are called cytoplasm, where many organelles are inside. So now let's say this this is the cell that is made up of over 70% water. So we take this cell and dip it into this solution that is hypertonic. What do you think will happen to this solution? Remember, I told you that hypertonic solution, they have higher concentration. All right, higher concentration than the concentration of the cell. Do you understand? So now there is a very important biological process that occurs in cells, and that process is called osmosis. which is the movement of liquid. Are you there? The movement of what? Liquids. All right, from a region of lower concentration to a region of higher concentration through a semi-permeable membrane. So there's liquid inside this cell. You take it and pour it. You take this cell, put it into the solution. So what happens? What did I say? Osmosis. You are going from lower to higher concentration. So remember, hypertonic solution have higher concentration, and this is low. This cell have lower concentration because I said hyper they have higher. I'm going hyper high. So you take this cell, put it into this solution. What do you think will happen when the cell enters into this solution? The water content in this cell start moving out from the cell. Do you understand? It start moving out from the cell into the solution because it's going from where a lower concentration region to a region of higher concentration. This osmosis. And what solution can cause that to happen? It is hypertonic solution. Hypotonic solution cannot. Do you understand what I'm saying? So all of these things must be noted. So when the water content of this cell has spilled out from the cell into the solution, into the hypertonic solution, into the container, what happens to this cell? It becomes shrinked. Do you understand? Shrinkage will occur. That is why the answer is hypertonic solution. I believe you understand what I'm saying. So for hypotonic solution is the direct opposite of what I just said. For hypo, let's say this container now is made up of hypotonic solution where there is low concentration. All right, so the cell's concentration is higher than that of the the solution's concentration. What happens following osmosis? The solution, basically the content of the solution will go from lower concentration to higher because now this solution is hypotonic. I believe you understand what I'm saying. All of these things are very simple. So this solution is hypotonic. So it's following osmosis which goes from lower to higher.
Alright, so moving further, we already know that cells are broadly divided into two groups, which include plant cells and animal cells. Now, let's take an instance. Let's talk about plant cells. Listen carefully. Now, we've established that whenever a cell at all, be it that cell is a plant cell or an animal cell, is placed in an hypotonic solution, what happens? We already know that an hypotonic solution has low concentration. Then the concentration of that cell is already higher than the concentration of that hypotonic solution. And following the process of osmosis that has to do with the movement of water, the movement of liquids from a region of lower concentration to a region of higher concentration, what happens to that cell when it is placed in an hypotonic solution? Since now the hypotonic solution has lower concentration, the water in that cell tends to flow into the cell because now the cell has higher concentration. I believe you are following me. So if you observe, the content of that solution start entering into the cell because it's following the process of osmosis, moving from lower concentration to higher concentration. What then happens to that cell? So let's explain for both plant cells and animal cells because it's very important for us to be specific. So let's talk about plant cells. Remember, plant cells have rigid cell wall and animal cells do not have that. Do you understand? So let's say that plant cell, because we're on plant cells now, plant cells start taking in this liquid coming from the hypotonic solution, taking it into the itself. You understand? What do you think will happen to that cell? Do you think that cell will burst? No. Plant cells actually do not burst. What happens to them? They will just be swollen. They will not burst. Why? Because of the presence of that rigid cell wall. So what we describe that is that we'll now say that that plant cell is turgid. Are you there? Please take note. Plant cells don't burst. So when they are placed in hypotonic solution, they take water to be swollen and then they become turgid. Are you there? There will be a great internal
external pressure found in them, hence they become turgid. Are you there? But now let's say this same plant cell was placed in an hypertonic solution. Remember, hypertonic solution have higher concentration than the cell's concentration. So now the cell's concentration, which is the plant cell in this case, have lower concentration. So what will happen? Remember, osmosis, the liquid moves from region of lower concentration to region of higher concentration. So when this plant cell now, because they have rigid cell wall, they are placed in hypertonic solution, the liquid in them start spilling away from their cytoplasm and then flow into the hypertonic solution. So when all the liquid content in their cytoplasm has actually moved out from the cell, I believe you know that plant cells have rigid cell wall. So when they lose all the liquid content in them, they shrink, and this process is called plasmolysis. Please don't mistake a cell being turgid and a cell undergoing plasmolysis. I believe you are following me. So these are the concepts of plant cells when placed in an hypotonic solution and as well as in an hypertonic solution. But now let's talk about animal cells. Now, first of all, let's talk about an animal cell placed in an hypotonic solution. I believe me saying this, you already understand the picture of what is happening because I've explained to you the concept. So this is an animal cell. Remember, animal cells, they don't have rigid cell wall. What they have is called cell membrane. Are you there? So when they are placed in an hypotonic solution, remember, hypotonic solution have low concentration than the cell's concentration. So the concentration of the cell is higher than that of the solution. So what happens? Remember, osmosis, it moves from region of lower concentration to region of higher concentration. So the content of the solution start moving to the cell because the solution has lower concentration. So it start moving into the cell. So what happens to this cell? This cell, because of, animal cells don't have rigid cell wall, so they tend to burst when they've absorbed so much water, when they've actually absorbed so much liquid. So when they actually absorb so much water and they've reached their limit, they tend to burst. And this is called cytolysis. Please take note, lysis means splitting. Are you there? Like the cell cytoplasm is already splitted, so it's called cytolysis. But now when this same animal cell is placed in an hypertonic solution where it moves from, following osmosis, the water basically moves from a region of lower concentration to a region of higher concentration. Now the cell now have lower concentration because it's placed in an hypertonic solution. The word hyper means high concentration. Then what happens is that the content of the cell start to enter into the solution, you understand, into the container. So with this, the cell start to shrink. Are you there? So when the cells start to shrink, we call it shrinkage. And this basically is explained by a process called crenation. Are you there? So please take note of what I just said. It's very, very important. So all these must be noted. All right, they are all very easy. It's very important for me to explain it this way so you'll be able to understand every single concept. All right, so let's move on. So too much solution of the hypotonic solution entering into the cell because it's going from lower to higher now. Because when you're talking about hypotonic solution, the concentration of the cell will be higher than that of the solution. So following osmosis, which moves from lower concentration to higher concentration, the cell will get more water and burst. But if it's an animal cell like red blood cell, we call it hemolysis. Do you understand? So all of this must be noted. So moving further, let's get into iso. From the word iso, it means the same. It means that the concentration of the container of the solution and the concentration of the cell, they are the same. So there will not be movement. Let's say this is an isotonic solution now, isotonic solution. What happens? Isotonic solution. What do you think will happen? They have same concentration, there will not be movement. There will not be any movement between the cell content and the solution's content. So the cell will just dead, nothing happens to the cell. But moving over to deionized water, deionized water works the way hypotonic solution is. It's very simple. What's deionized? All right, you know, when water is ionized, it means that it has so many ions inside. But now it is deionized, no ions inside. What do you think is happening? It's very simple. So since there is no ion inside, then that water tends to be hypotonic. That water's concentration will decrease because you've removed so much ions inside. You can say that the water is deionized. So many ions have been removed from that water. So making the concentration of that water to be reduced. Do you understand what's happening? So when it's reduced, then it's going to work the way hypotonic solutions are working. I believe you understand the picture of everything. So you can see we've analyzed this question and we've gotten the answer to this question. Let's get into the next practice question. And the question says, question 3, it says, which of the following is true of leukocytes? All right, from the look of things, this question is on blood. Now, you must know here that the blood is a connective tissue. You can see the word connective. It is made up of cells. For you to be a tissue, you'll be made up of cells, and all of these cells are connected together. Now, listen carefully. You must understand here that the blood basically is made up of different cells, which include the blood, different contents rather. We have blood cells, generally blood cells, and as well as plasma. But what I'm stressing on is the blood cells. Now, blood cells can be either white blood cells, WBC, we have red blood cells, ROBC, and as well as we have what we call thrombocytes. All right, thrombocytes. If we don't call them thrombocytes, we call them platelets. What did I call them? Platelets. Platelets are also called thrombocytes. They are involved in what? Blood clotting. In case, let's say for instance, a particular individual got injured and there is bleeding, these platelets, also called thrombocytes, will be released and go basically to that injury site, then to stop the bleeding. That's the function of thrombocytes. Well, red blood cells basically, they help to carry oxygen. Do you understand? There is a pigment found in red blood cell. They call that pigment hemoglobin. Do you understand? They help in transport of oxygen throughout the body. Do you understand? That pigment is also called a respiratory pigment. But white blood cells basically, they help to fight against infections, fight against disease. So white blood cells basically, they help the body for immune response. They help the body's immunity. Do you understand? To fight against other invaders. So this must be noted. White blood cells are also called leukocytes, all right, where the red blood cells are also called erythrocytes. Now, in size, white blood cells are larger and they contain nucleus. Do you understand? But red blood cells are smaller with no nucleus. Most especially when these red blood cells are matured, they lack nucleus. Do you understand? So I believe I've explained the overview. So let's solve the question. Which of the following is true? I've said many things about all of these things. So let's learn which of the following is true about leukocyte, about these white blood cells. Number 1, option A, they are large and they are nucleated, meaning that they have nucleus. Wow, that's the answer. Option B, they are involved in blood clotting. That is false. What is involved in blood clotting? It is thrombocytes, also called platelet. Moving further, option C, they are respiratory pigments. None of the cells are respiratory pigments. The pigment I just mentioned initially was hemoglobin, and that pigment is associated with the red blood cell. It has no business with white blood cell. All right, I told you the function already. And option D is none of the above. So what becomes the answer to this question? It is option A, because white blood cells generally are large and also they have nucleus in them. They are nucleated. Let's get into the next practice question, which is question number 4. It says, the ability of a living organism to detect and respond to changes in the environment is referred to as what? Now, listen carefully. Listen. The answer to this question is irritability. Irritability is also called sensitivity. That's the ability of an organism not just an organism like humans, we humans are organisms, to be able to detect and respond to changes in the environment. Let's say, for instance, you change the environment, and the environment you are now in is an environment that is cold. What do you think will happen? Definitely, as soon as you get to that place that is very cold, your body system will detect that it's very cold and try to respond to the changes in the environment. At first, you might not be able to, adapt. Do you understand? But as time goes on, you adapt, and that explains irritability. That does not explain locomotion. Locomotion is just where an organism moves from one point to another. Do you understand? Taxis is basically a directional movement towards a stimuli. Taxi is what? A directional movement towards a stimulus. Do you understand? For instance, taxis is divided into two. We have positive and negative. So when that stimuli is elicited, all right, when that stimuli is initiated,
there are various examples of stimuli that can actually, initiate this directional movement, which is called taxes. Now, this must be noted. Before we progress, it is important for us to understand what stimuli talks about. Now, stimuli is basically anything that can cause change in an organism. Are you there? Be it physical change or behavioral change, is called a stimuli. Are you there? So anything that can cause change in an organism, be it physical or behavioral, we call it stimuli. So let us progress. All right, so please listen carefully. One of them is light. When an organism actually moves towards light, in response to light. All right, let's say there is light and the organism tends to move towards that light, we call that type of taxis phototaxis. Are you there? Because it has to do with light. But now we have other examples of stimuli, we have other examples like chemicals. When an organism actually moves towards, the presence of a chemical, that is called chemotaxis. Are you there? When that same organism actually moves towards in response to, let's say, gravity, we call that type of taxis geotaxis. Are you there? So we have various examples of these taxis, which is a directional movement of organisms towards a stimuli. So I believe now all of these things must be noted.
So that's how all of this works. So the answer can be this, the answer can be this, and the answer basically is irritability. The answer to the question can be growth, because growth is an irreversible increase in mass, size, and complexity of a living organism. The organism will increase in size, will increase in mass, and will increase in complexity in terms of the way the body is built. Do you understand? In way the cells and tissues and organs are arranged. So growth is basically an irreversible increase in mass, size, and as well as complexity of a living organism. So let's get into the next practice question.
To have more understanding about chemistry calculations and solving all of these type of questions, I already have them on the chemistry masterpiece, okay, which is calculations and keynotes in chemistry. So if you are preparing to write the Jamb exam, this book is very, very important for you. Now, for more information getting the chemistry masterpiece, do well to direct message me via my WhatsApp number. It will be on the screen.
Okay guys, let's get into the next practice question, and it says the vector of plasmodiasis is called. Now this question is very simple. You must understand here that plasmodiasis is also regarded to be called malaria. So what is the vector for the disease called malaria? It is simply called the female Anopheles mosquito. All right, the female Anopheles mosquito. You must understand here that Plasmodiasis is also called malaria, and the vector that actually carries the pathogen that causes the disease called malaria or plasmodiasis is the female Anopheles mosquito. Are you there? The Anopheles mosquito is of two species. We have the male and the female. The female feeds on blood. Are you there? Whereby the male feeds on plant juice. The female feeds on animal juice, and the animal juice here is blood. Are you there? So this must be noted. All right. The type of asexual reproduction in Paramecium is called, the answer is simply called binary fission. Binary fission, whereby if it's to reproduce paramecium, we actually split into two. Binary means two, fission means splitting. Do you understand? So basically, you must understand here that paramecium is a protozoa. It belongs to the kingdom Protista. Remember, kingdom Protista is the kingdom of classification, and it is broadly divided into two groups, which include protozoa and protophyta. Protozoas, they are the animal-like protists. Protophytas, they are the plant-like protists. Are you there? So this must be noted, whereby Paramecium actually is a protozoan. Do you understand? It is an animal-like protist. It behaves like an animal, and they reproduce by binary fission asexually. But also Paramecium also reproduce sexually, and if Paramecium is to reproduce sexually, it reproduces by what we call conjugation. What we call what? Conjugation. So this must be noted. Are you there? So let's get into the next practice question, and it says the instrument used to measure relative humidity is called hygrometer. Remember, relative humidity is an abiotic consumer of the ecosystem. Are you there? So this must be noted. We have other abiotic components which include temperature, which is measured by the thermometer, which include light, which is measured by what we call photometer, which include soil moisture, and this is measured by what we call the tensiometer. Are you there? We have other various instruments like wind speed, which is measured by anemometer, wind direction, which is measured by wind vane, rainfall, which is measured by the rain gauge. Are you there? All of these are examples of abiotic component. Abiotic component of the ecosystem doesn't have life in them, but for the biotic component, they do have life in them, which include also decomposers. Are you there? All of these are the biotic component of the ecosystem, and as well as. So moving further to the next question, question number 8, it says exoerythrocytic phase of the life cycle of malaria parasite occurs where? It occurs in the liver. What this question actually wants us to understand is that they are actually asking us for the life cycle of the malaria parasite. How this malaria parasite, which is called the female Anopheles mosquito, actually passes the pathogen into the individual to cause malaria. Are you there? So that's what this question says. But take note, the exoerythrocytic phase. Exo means outside the erythrocytes. Do you understand? So let me explain how it works. The way it works is this. The life cycle of malaria. The female Anopheles mosquito actually first of all bites the individual. When the female Anopheles mosquito bites the individual, it injects what we call sporozoites. Are you there? It injects sporozoites into the bloodstream of that individual. Then that sporozoite tends to grow and also flow towards the liver. As soon as the sporozoite enters the liver, it changes into what we call trophozoites. Then the trophozoite further grows and forms what we call merozoites. Are you there? So if you observe what I just said, first of all, the female Anopheles bites and inject what we call sporozoite. The sporozoite basically, grows and multiply and flow towards the liver and forms what we call trophozoite. Then the trophozoite forms what we call merozoites. Are you there? So if you observe, as soon as the trophozoite in the liver now then forms what we call merozoite, you can see here that the pathogen basically is now in the liver. So as soon as it gets to the liver, after the liver, it tends to go to the red blood cells specifically. So that's why it's called exoerythrocytic. Exo means, outside the red blood cell. So basically, when it enters the liver, it's still outside the red blood cell. Then it enters the red blood cell. And when it enters the red blood cell, basically it should be called erythrocytic phase, not exoerythrocytic phase. I believe you understand what I'm saying. So just understand that the exoerythrocytic phase of the life cycle of malaria occurs in the liver, but as soon as it enters the red blood cell, it's not exoerythrocytic phase rather, but rather erythrocytic phase itself, whereby the parasite now then degrades the red blood cell to actually cause the disease malaria. Are you there? So all of these must be noted. All right, moving further, question number 9 says habitats are generally classified into 3 major groups, which include the aquatic habitat, which include the terrestrial habitat, and also the arboreal habitat. You understand? So moving over to the next question, which says the first organism evolved from which habitat? Actually, is called the aquatic habitat. We have so many organisms in water. Organisms, now the way it works is this. It's very funny because organisms you see on land, most of them are also found in water. Like they are also in water. Like the one I'm trying to say here is this. You have horse. You understand horse on terrestrial habitat on land. We also have what we call sea horse in water. We have lion. We also have what we call sea lion. Like we have so many organisms that are found in water. So the first organism evolved from the aquatic habitat because we have so many types of organisms in water. Do you understand? Please take note of what I just said. So the next question says, what is hypogeo germination? This is a type of germination whereby the seed leaf of that particular seed, seed leaves are also called cotyledons, grows beneath the ground, under the ground, before it now comes up. Just a hypogeo, under. The cotyledons start growing under before it comes up. That's hypogeo. But epigeo, the cotyledon with the seed leaves grows, above the ground level. Do you understand? That's how it works. We have examples of, plants that undergo hypogeo and as well as epigeo germination. For hypogeo, we have examples of plants which include beans, which include sunflower, which include cotton, which include pea, which include groundnut. All of these are examples of plants that undergo hypogeo, but plants that undergo epigeo include tomatoes, which is the Lycopersicum esculentus, which also include cucumber. Do you understand? I said Lycopersicum esculentus is the botanical name for tomatoes. Are you there? So all of this must be noted. Moving over to the next question, question number 13, it says, in vascular plants, xylem tissues are responsible for what? These xylem tissues, they are the vascular bundle that supports the plant in transport of nutrients from the soil, you understand, to the leaf, to the plant in general. So basically, it not just transport nutrients, it also transport water. You understand? So basically, the answer to this question should be transportation of water, not storage of sugar, not transportation of hormones. Are you there? Not transportation of hormones, actually. Now, this must be noted. Majorly, it must be noted here that the structure in plant that helps to transport hormones more is the phloem tissues. Are you there? Phloem tissues. Though the xylem also involve, but most of the hormones, which include auxin, gibberellin, abscisic acid, cytokinins, all of these are plant hormones. Majorly, they are transported by the phloem tissues in plant, not the xylem, but xylem also are involved. But majorly, xylem function is to transport water and nutrients. Are you there? So let's get into the next question, which says, the causative agent, all right, of tuberculosis is basically what actually causes tuberculosis. The causative agent or pathogen, causative agents are also called pathogen, is called Mycobacterium tuberculosis or tuberculosis. All right, Mycobacterium tuberculosis or tuberculosis. Are you there? So all of these must be noted. In some other questions, the options might be the causative agent for tuberculosis is A, bacteria, B, virus, C, fungus, D, none of the above. What becomes your answer? The causative carrier agent for tuberculosis is a bacteria. How did I know? From the exact causative agent called pathogen, you can see there's bacterium there. So actually, tuberculosis is a bacteria type of disease. Are you there? So all of this must be noted. It can be gotten by coughing, sneezing around a person that is infected. Do you understand? So tuberculosis is what? Airborne. Take note. So moving further, the next question says, which of these is a respiratory organ in mammals in we humans majorly? All right, it is the lungs. The mouth is not primarily involved in respiration. All right, though we use our mouth to breathe in a way. Do you understand? But majorly it is done by the lungs. Do you understand? So the lungs is the chief respiratory organ. For heart, it controls circulation of blood. Mouth controls injection of food. Do you understand? And we have various accessory organs in the mouth, which include the teeth for chewing food and the rest, tongue, swallowing and the rest. So skin does not help in respiration, it helps in excretion and protection. Do you understand? And other functions, all right? So this must be noted. Moving over to the next question, it says the central nervous system comprises of chiefly the brain and the spinal cord. Take note. The central nervous system is made up of what? The brain and the spinal cord. We have the nervous system, which is divided into two other systems, which include the central nervous system and the peripheral nervous system. The peripheral nervous systems majorly explain neurons, nerves. All right, but in more video lessons on my channel, I'm going to deeply explain all of these systems. So make sure you watch out for those lessons. Moving over to the next question, which says, in glycolysis, glucose is broken down to, all right, glycolysis is a type of reaction that occurs in a type of respiration called aerobic respiration. Aerobic respiration basically is a type of respiration that occurs in the presence of oxygen. Respiration are of two types majorly, all right, in terms of oxygen, all right, because we have internal and external. But in terms of oxygen, respiration are of two types. We have aerobic and anaerobic respiration. Aerobic occurs in the presence of oxygen. Anaerobic occurs without oxygen. So on aerobic respiration, we have two types of reactions. The first one is glycolysis and the other is Krebs cycle. So they are not asking us what glycolysis entails. Now glycolysis is a breakdown type of reaction. So what will you call glycolysis? Is it a catabolic reaction or an anabolic reaction? I said you can also see the word breakdown. So any reaction that deals with breakdown of a substance is catabolic, but any reaction that deals with build up of a substance is anabolic. And a good example of an anabolic reaction is photosynthesis. Are you there? Photosynthesis is anabolic because it's a build-up process. Respiration generally is catabolic. Are you there? So glycolysis will be catabolic. You can even see from the word breakdown. Glucose is actually broken down to a 3-carbon compound called pyruvic acid, and as well energy is released, which is called ATP. But there is a proper reaction to be written, but this must be noted. Remember, ATP is called adenosine triphosphate and it is the energy currency of the body. Are you there? So let's get into the next question. And the next question says, the muscular sheath that separates the thoracic, these are thoracic, the chest region, the thoracic region, from the abdominal, the abdomen, where the stomach is found. Are you there? So then that's what separates, there's a muscle that helps in respiration that separates the thoracic from the abdominal cavity. That muscular sheath is called the diaphragm. All right, so this must be noted, diaphragm. So this must be noted. Let's get into the next practice question. Okay guys, let's get into the next practice question, and it says, in mammals, in humans, all right, the gland which performs both endocrine and exocrine function is, the gland is called the pancreas gland. Please note this. Please, the pancreas gland is a dual gland. It is a gland that performs two functions, all right? We have different types of glands, majorly endocrine and exocrine. What are endocrine glands? They are glands that basically secrete hormones, whereby exocrine glands are glands that don't secrete hormones, and now listen, endocrine glands are ductless glands. What they secrete goes directly into the bloodstream. And what did they secrete? I just said hormones. But these, they secrete all other substances in the body that are not hormones. Are you there? and for them to be secreted, they will follow through a duct. There will be a passage for them to be secreted, and that makes them to be a duct gland. Are you there? So these are ductless and these are duct gland. Pancreas, how is pancreas an endocrine gland? Is an endocrine gland because it produces hormones. And what are the hormones that this pancreas gland produces? They are insulin and glucagon. But insulin and glucagon specifically are produced by a cell inside the pancreas gland, and that cell is called the islet of Langerhans, which basically produces what we call the beta and alpha cells. Beta cells produces insulin, alpha cells produces glucagon in the pancreas gland. The pancreas gland also serves as an exocrine gland because it produces digestive enzymes, which include amylase, protease, and lipase. Amylase acts on carbohydrates, protease acts on proteins, lipase acts on lipids, also called fats and oil. So that's the answer to this question. Question number 20 says, the chitin in exoskeleton of arthropods are strengthened by calcium compounds. Please take note of it. This is the answer to the question. Chitin are found in exoskeleton, that's the body of arthropods. Remember, arthropods are part of the invertebrates. We have examples which include the poriferas, the colenterata, the platyhelminths. That's what we call them, the platyhelminths, helmet worms. Are you there? We have the annelids, we have the arthropods, we have the molluscas. Are you there? So all of these are examples of invertebrates, but we have vertebrates as well. All of these are under the kingdom Animalia on classification. In subsequent video, I'm going to talk more about all of these, but take note of what I just said. The exoskeleton, the chitin in the exoskeleton of arthropod is strengthened by what? Calcium compound. So this must be noted. Moving over to the next question, and here is the question. They just brought out the structure and they said we should label the structure. From the look of things, this structure is called the nephron. Remember, the nephron is the basic functional and structural unit of the kidney. All right, these nephrons are found inside the kidneys. All right, we have about 1 million nephrons in one kidney. Now we have two kidneys, so approximately we have about 2 million nephrons. All right, if these nephrons are damaged in a particular kidney, the kidney is almost useless. So it's important for us to understand the function of the nephron and as well as understanding the parts of the nephron. The nephron has two major parts, which include the renal corpuscle, all right, renal corpuscle, and as well as the renal tubule. Now, if you observe, I said renal. Anytime we talk about the kidney, we use the word renal. You understand? So don't get confused. Renal is the kidney. Hepatic is the liver. So moving further, I said it's divided into the renal corpuscle and the renal tubule. The renal corpuscle has two parts, which include the glomerulus, all right, the glomerulus, and also the Bowman's capsule, all right, the Bowman's capsule. This must be noted, all right, whereby the renal tubule is made up of different parts. They are tubes, so that's why we call them tubules. So basically, just listen carefully. This up part is the renal corpuscle, and it's made up of this thing that are networks of capillaries. We call them glomerulus. Are you there? So basically, we are answering all of these questions from question 21 to question, all right, 26. Are you there? So listen carefully. So if you observe on description, this is a network of capillary, all right. It takes blood from what we call the afferent arterioles. But just understand that this is glomerulus and it's what? A network of capillaries. Note that. Whereby this thing here that covers the houses the glomerulus is called the Bowman's capsule. All right, the Bowman's capsule. Do you understand? So all of these must be noted. Moving further, you can see all of the remaining parts are like tubes. This first part, we call it the PCT. All right, what's PCT? Proximal convoluted tubule. Proximal convoluted tubule. So on labeling, we already labeling the part PCT, proximal convoluted tubule, whereby this other part, this one that comes down, is called the loop of Henle. All right, they call it the loop of Henle. LOH, loop of Henle. Are you there? So this must be noted. Moving over to this other part, we've described this to be loop of Henle. This one is called the DCT, which is called the distal convoluted tubule. Distal is away. Distal means away. All right. Distal convoluted tubule. You there? So this must be noted. Whereby this other part that comes down, all right, is called the collecting duct. Are you there? It's called what? The collecting duct. So these are the parts of what now? The nephron. This must be noted. So we've answered all of these questions from question 21 to question 26. So question 27 says the membrane of the lungs is called the pleural membrane. All right, it's called the pleural membrane. Take note, this is the membrane that covers the lungs. The membrane that houses or covers or bounds the heart is called the pericardium. Are you there? So all major organs in the body have their respective membranes. The membrane for the lungs is called the pleural membrane. For the heart is called the pericardium, or we simply call it the pericardial membrane. So moving further, the next question says, which gland produces hormones and enzymes? I've done the explanation above. I will not put the answer. Make sure you put the answer to question number 28 in the comment section. I've explained the concept already. There is a gland in the body that produces hormones and as well as enzymes. So make sure you provide the answer. I have to write out the question again in another format so you can answer the question. So make sure you provide the answer to this question. So moving over to the next question, it says, a DNA molecule is made up of what? All right. DNA molecule is made up of what we call sugar. All right. Sugar, but specifically a 5-carbon sugar, which is called the pentose sugar. All right. It's made up of what we call nitrogenous bases, and a special group called the phosphate group. So all of this must be noted about DNA. All right. So take note, DNA has a double helical structure. A double helical structure. This is how it is. Do you understand? It's a double helical structure. The DNA molecule has a double. If you look at it, if you want to stress on the DNA, we actually show all of these groups: sugar, nitrogenous base, and the phosphate group. But please just note what the DNA is made up of. Moving further, let's get into question number 30, and it says the gland in the eyes that produces tears is simply called the lacrimal gland. This must be noted. All right, so let's get into the next practice question. All right, you can see two diagrams on the board, so we'll be analyzing them. So listen carefully. All right, so let's analyze this first diagram. All right, now looking at this diagram explains the urinary system, the system in the body that controls urine production. Are you there? So the diagram represents the urinary system. Are you there? So that is for that. Moving over, let's talk about the components of the urinary system. First of all, the urinary system is made up of the kidneys. Are you there? Now, remember, what's the function of the kidney? One of the major functions of the kidney is to filter blood. So what actually filters this blood is the nephron in each of the kidney. Remember, when we stand anatomically like this is the left kidney, and also this is the right kidney, because we need that in this question. So where do you know that this is the kidney, but which is the left and which is the right? Anatomically, this is how you should stand. So this is left. So Roman figure 2. All right, so here they say that the part labeled this is the left kidney. All right, whereby this part that is labeled Roman figure 1 is the right kidney. Are you there? So this must be noted. Moving further, after the kidney actually takes the blood and is filtered inside this structure, inside the kidney, then the waste products, which one of them is urine, then goes through these structures. These are called ureters. Please, they are not called urethra. This is the urethra. Are you there? So it is the ureters. There are two ureters that takes the urine into what we call the bladder. So this structure here is called the bladder. When the bladder is filled, it then passes the urine through the urethra and then passes the urine away. So This is how the urinary system works. Blood is filtered, then goes to the ureter, to the bladder, the urethra, and passes out as waste products. Are you there? So all of this must be noted. So you can see we've analyzed every single part of all the questions. All right, all the parts they say we should label, we've labeled them. So that is for the urinary system. Are you there? So let's go into this other part. This question. Now, this structure we are seeing on the board represents what we call the biliary tree. All right, we call it the biliary tree or the biliary tract. Do you understand? Now, listen. The biliary tract has so many important organs, which basically include this structure we are seeing here is called the liver. Are you there? So this must be noted. So the diagram represents the biliary tract, whereby this one that has like a J shape is the stomach. This is the stomach.
Make sure
whereby
you click on the subscribe button.
these other structure, let's try to label each of them. Now you can see after the stomach there is another one that comes like this. This is called the small intestine. Remember, the small intestine has three parts. All right, the ileum, the duodenum, and the jejunum. Are you there? So this other structure you are seeing is the pancreas. You know the pancreas. We've been talking about it in the course of this lesson. So this is the pancreas. Moving further, which part have I not labeled? I've labeled the liver, I've labeled the stomach, I've labeled the pancreas. All right, this thing here is simply called gallbladder. This must be noted. So all of this forms the biliary tract. We've come to the end of this video lesson. If you find my video lesson helpful,
to have more understanding about chemistry calculations and solving all of this type of questions, I already have them on the chemistry masterpiece, okay, which is calculations and keynotes in chemistry. So if you are preparing to write the Jamb exam, this book is very, very important for you. Now, for more information getting the chemistry masterpiece, do well to direct message me via my WhatsApp number. It will be on the screen.